Good evening, friends. Today we are going to discuss a topic which is both clinically important and also important in terms of exams called myocardium. But in this uh, lecture, we won't be taking a typical textbook approach. We'll look at myocardium from a fresh perspective, from an evolutionary approach. And this will make the whole thing very interesting. It's going to stimulate our overall interest in the topic. And that's why I've taken the initiative of looking at the myocardium from overall a new perspective, and this will kind of stimulate interest. And let's start with the whole thing. Myocardium is a remarkable tissue. It beats around 3 billion times in our whole lifetime, and we are hardly conscious of its contraction. And that sounds true for most of our body. Our lungs are continuously inhaling and exhaling air, and we are hardly conscious of it. Our intestines, they are continuously contracting, relaxing, pushing the bolus of food ahead, and we are hardly conscious of it. If someone asks from us, where exactly is the seat of intelligence in your body, then we will say, yeah, brain. Brain is the seat of intelligence in our body. But our body has multiple layers of intelligence. And this is just one form of intelligence which our body has got. Even our intestines, they are quite an intelligent organ. Whenever a bolus of food goes into intestine, the front part of intestine relaxes, back part of the intestine contracts. There is a question in this previous NEET exam about what are the chemicals which are responsible for this contraction and relaxation. Now you see, intestine is a supremely intelligent organ because we have got sensors inside the intestine. They sense the acidity, alkalinity of the food, and then we've got interneurons which process this whole neural information, and then this information is processed, and according to that the enzymes are released. According to that the intestine relaxes and contracts. There are interneurons which control the rate at which the food will be emptied from the stomach. There are interneurons which decide how much should be the alkalinity and acidity. It's a supremely intelligent organ. There are many more intelligent organs. Even myocardium is a highly intelligent organ. Whenever we exercise, metabolic needs of body rise. The heart rate of myocardium increases. Then there are many myriad other factors ranging from thyroid hormone to progesterone which exert its effect on heart. So there are many intelligent systems within our body continuously interacting with each other, right? Now, how come this local intelligence came into being? Whenever we are just conscious of one form of intelligence, how come this local intelligence came into being? We will go very briefly into the evolutionary part of it. Initially, we had just one cell, unicellular organisms. Gradually, these cells started living together, and what we got is a form of multicellular colonies. Now these multicellular colonies gradually distributed the labor amongst themselves. That means the outer layer cells will do protective functions, will do locomotive functions, and the inner layer of cells will do some kind of digestive functions, right? Later on, these multicellular colonies developed some kind of gut within, and we got something like this. The cells in the immediate vicinity of it have to perform some kind of absorptive function, and the cells over here have to perform some kind of digestive function, and the cells on the periphery have to perform some kind of protective function and locomotive function. But all these functions need to be coordinated, and certain cells are specialized for managing this coordination. And this is how the first nervous system was born. So when the first nervous system was born, there was no brain. The nervous system was born just for better coordination amongst the cells, and it is still doing the same thing through the rest of our body. I mean, our body has many local centers of intelligence ranging from our intestine to our liver to our heart. Each part of our body is locally very intelligent. I mean, there is no need for a central processing unit for survival. I mean, if you're interested in evolution, we can take one briefly. We can take a very important, a very curious example of jellyfish. The jellyfish has got no central processing unit. It has got around 28 to 29 different ganglia. And each of these ganglion processes information and makes its own decision, and still the organism survives. Still this organism, jellyfish, is very successful. It can move towards light, it can hunt for its prey, it can reproduce, it can do everything despite having no central processing unit like brain. Each of its ganglia can make its own local decisions. And our body is still now, we are not very much different from jellyfish. We have got a central processing unit, but each part, each region of our body still makes its own decision based on its own neural processing, and that thing helps to keep us alive. So physiology is very interesting if you just look at it from evolutionary point of view. Now coming back to myocardium proper. I may have gone a bit off the topic, but this was just to make the things a little interesting. Now when we talk about myocardium, we know that we have got two kinds of two kinds of autonomic nervous system acting on our heart. One is parasympathetic nervous system, another is sympathetic nervous system. Whenever we say sympathetic and parasympathetic, it makes us feel as if both the nervous system are equal and well balanced. I mean, it feels as if both the nervous system are quite equal in strength, but this is not the case. Parasympathetic nervous system dominates the sympathetic nervous system, especially in the case of heart. The vagal stimulation, the strength of vagal stimulation is way stronger than sympathetic stimulation. So, I mean, if we take out our heart and keep it in some kind of petri dish which gives its glucose, ATP, everything, then will it beat or not? Of course it will beat, because heart beats because of its own intrinsic rhythm as a node. And what will be the heartbeat? The heart will beat at much faster rate. It will beat at around 120 beats per minute. It will be a lot more uninhibited because of the lack of vagal inhibition. So it's basically our vagal tone which inhibits the heart rate all the way from 120 to 72 beats per minute, right? Now we can do various kind of experiments on heart. I mean, of course not in humans, but on frogs and etc. And we can come to know myriad interesting properties of heart. One of the interesting properties of heart is vagal escape, right? So what exactly is vagal escape? Now, if we stimulate the vagus nerve, of course, the heart rate will go down. If we stimulate increase the strength of vagal stimulation, the heart rate will go down further. If we further increase the strength of vagal stimulation, the heart will stop. Right? Now, what exactly will happen if we keep stimulating the vagus nerve? Now, any mature person will stop at this point. You want to stimulate the vagus nerve further once the heart stops. But in science, you have got to be like kid. You have got to be curious like a kid. So if you keep on stimulating the vagus nerve even after the heart has stopped, then one paradoxical thing is going to happen. And that paradoxical thing is that the heart will start beating again on its own, right? If you keep stimulating the vagus nerve, the heart will start beating again. To diagrammatically represent it, I'll just draw it over here. Suppose this is our pulse. Now we started giving vagal stimulation. First of all, mild. If we give mild vagal stimulation, of course, the heart rate will fall down. Right? If we give mild vagal stimulation, heart rate will fall down. If we give a further strong vagal stimulation, the heart rate will completely stop. Heart rate will completely stop. But after some time, if we keep giving this vagal stimulation, then the heart will start beating again. So why exactly the vagal escape happens? Why exactly the heart starts beating on its own after continuous vigorous vagal stimulation? For understanding this, we need to understand a very interesting property of cardiac tissue, and that property is that every part of the conductive tissue of heart is a potential pacemaker. So all the conductive tissue of heart, ranging from atria all the way up, ranging from SA node all the way up to AV node, bundle of his Purkinje fibers, each point in this is a potential pacemaker. Now, then why exactly SA node supersedes over everything? Because SA node beats with the highest frequency. So any point which beats with the highest frequency supersedes over the other points. So this is the reason that the whole of heart beats with the frequency of the SA node. Now, in this case, we are stimulating the vagus and inhibiting the SA node. Once the SA node is inhibited, the other points which up till now were under the influence of the rhythm of SA node will get free, will get released. So they get stimulated and they start generating their own impulse. And this phenomena is called idioventricular rhythm. So idio, because we don't know where exactly in the ventricles this impulse is coming from, and ventricular because we definitely know that this is somewhere in the ventricles. Idio literally means we don't know. Idiopathic is a very important word in medical vocabulary. Idiopathic means something like we don't know the exact cause, like most common cause of diabetes, idiopathic. Most common cause of hypothyroidism, idiopathic. Most common cause of hypertension, idiopathic. So idio means we don't know. If pathic comes, we don't know the cause. Here, idioventricular, we don't know the exact site from where this impulse is coming, and ventricular means we definitely know it is coming somewhere from the ventricles. There are other hypotheses for vagal escape, like depletion of acetylcholine. When you continuously stimulate the vagus nerve, the acetylcholine inside reserves get depleted, and then further stimulation of vagus nerve does not produce any effect. Then there is one more theory called concomitant stimulation of sympathetic nerve. Now there is a huge difference between a frog's heart and a human's heart. In frog's heart we have got one ventricle, and in humans we have got left and right ventricle separately. Apart from that, in frog's heart, the vagus and sympathetic nerve, they combine together and supply the heart. So when in frog's heart you are continuously stimulating the vagus nerve, you are also side by side stimulating the sympathetic nerve. But initially the stimulation of vagus nerve supersedes over the sympathetic nerve, right? So once the vagus nerve get tired, stimulation of sympathetic nerve can restart the heart. So these are the three theories. Of the strongest is of course idioventricular rhythm. The second one is depletion of acetylcholine, and the third one is concomitant stimulation of the sympathetic trunk. Now, what are the other ways in which we can experiment with heart? I mean, we can put some cold saline and some hot saline on the frog's heart and just see how the frog heart react to it. And this thing is very interesting, and this thing really requires an in-depth understanding of physiology, right? Now, what happens if we cool something or heat something? For understanding what kind of effect the cold water or hot water is going to have on the frog's heart, we have to understand what these cold water and hot water, in what way they affect a normal skeletal muscle. For that we have to first of all draw a normal skeletal muscle curve. This is the point of stimulation, and this is the point of contraction. Right. This is the height of contraction. I know very well that this is the point of stimulation, this is the point of contraction. This means this thing is our latent period, and this thing is our contraction period, and this thing is our relaxation period. Latent period, contraction period, and relaxation period. Apart from that, there is something like this, which is a physiological curve because of the inertia of the lever, right? Now, what happens if we kind of heat the skeletal muscle, or put a hot Ringer's solution, put the skeletal muscle in a hot Ringer's solution? Then what exactly is going to happen? Then we get a graph something like this. Now, what we have observed when we put the skeletal muscle in a hot Ringer's solution, what exactly has happened is that latent period has shortened, the contraction period has shortened, the relaxation period has shortened, and the amplitude of contraction has increased. So when you heat something, everything speeds up. The movement of ions across the membrane speeds up, the viscosity of the muscle decreases, and all the chemical and metabolic processes inside the muscle speeds up. That's why you get higher amplitude of contraction and shortening of contraction and relaxation period and shortening of the latent period. Right? Now what will happen if you cool down if you put the skeletal muscle in a cool solution? Cool is, if you put the solution in a cool, then what you're going to find is like something like this. So what exactly has happened in this case is that latent period has increased and contraction period has increased and relaxation period has increased and amplitude of contraction has decreased. Now this means that when you cool down something, all the chemical and metabolic processes slow down, all the ion channels slow down, viscosity of the muscle increases, and even inertia of the liver increases. That's why you get this kind of graph. So this is the basic idea we have to keep in our mind when we experiment with the kind of frog's heart and cool it down or heat it up, right? So these graphs might come even in PG entrance exams and even in university exams, right? Now, if we put some cold water on the sinus venosus of frog. Now, frog is not exactly, in frog we have got one sinus venosus which is formed due to the fusion of the superior vena cava and inferior vena cava. And that sinus venosus drains into auricle and that auricle drains into ventricle. And the pacemaker of a frog's heart is basically sinus venosus. If you put few drops of cold water on sinus venosus, then what exactly you will expect, right? Now suppose this is a normal heartbeat of a frog. Now one peculiar thing about a frog's heartbeat is that we are going to have kind of mild connection over here, right? Typical thing about frog's heartbeat is that we are going to have 3 bumps, right? We are going to have first bump which is due to contraction of sinus venosus, second bump which is due to contraction of auricle, and third bump which is due to the contraction of ventricle, right? So this is normal heart cardiogram. Now if you put some cold water on the sinus venosus, then what you are going to expect, right? Now this is the trick I play. I always ask it from the students in my class, then what exactly you are going to expect if you put some cold water on sinus venosus. And most of the students in a reflex they answer that the amplitude of contraction will go down as soon as you put cold water on sinus venosus. Now this is quite intuitive also, right? Yes, if cold slows down everything, reduces amplitude of contraction, then as soon as you put some cold water on sinus venosus, the amplitude of contraction should go down. But that is definitely not the case over here. And for understanding it, actually the amplitude of contraction falls down, right? This is paradoxical. As soon as you put some cold water over here, the amplitude of contraction actually goes down, right? Amplitude of contraction goes down, but heart rate slows. So why exactly, amplitude of contraction will actually increase, right? Amplitude of contraction will increase, but the heart rate if you put some cold water, the amplitude of contraction is going to increase, but the heart rate will slow down. So why exactly the amplitude of contraction is going to increase if you put some cold water on sinus venosus, right? You have to think in a step-by-step manner why exactly this will happen, right? Now, as soon as you put some cold water on sinus venosus, the heart rate goes down. The heart rate goes down means the impulses are far apart from each other. The impulses are far apart from each other means that the ventricles will get more time to get filled. If the ventricles will get more time to get filled, this means Frank's Starling law. Force is directly proportional to length, right? So ventricles will contract more vigorously, and when the ventricles will contract more vigorously, the amplitude of contraction is going to increase, but the rate of contraction, because sinus venosus have been poured with cold water, rate of contraction will decrease. So this is the reason as soon as you pour some cold water on sinus venosus, amplitude of contraction will increase, but the rate of contraction will decrease. So this kind of graph can come in your exams, and they might ask what we have poured over here. This is cold water or hot water, right? So in normal skeletal muscle, if amplitude is increasing, that means hot water. But in case of sinus venosus or frog, if amplitude is increasing, this means cold water have been poured, right? Now what will happen if we pour some hot water? Hot water on sinus venosus. Now we have to think in a step-by-step manner and very logically over here. If we pour some hot water on sinus venosus, the heart rate will increase. And as soon as the heart rate increases, this means the impulses are closely packed with each other. And if the impulses are closely packed with each other, this means that the ventricles won't get enough time to get filled. So improperly filled ventricles, that means improper contraction. And improper contraction means less amplitude, right? There will be less amplitude of contraction, but these will be quite close to each other, right? Very close to each other. So there will be crowding of impulses, but amplitude will be less if you pour hot water on sinus venosus, right? Now coming to ventricles, right? Now what will happen if you directly put water on ventricles? One practical point over here is that when heating or cooling the ventricles, we soak the filter paper in water and then put it on the ventricles. This is to avoid seeping of water from the ventricles all the way up to sinus venosus, right? So what will happen if we directly heat or cool the ventricles? Now since sinus venosus is not being touched, that means the heart rate won't change. And the heart rate won't change means ventricle will behave just like the skeletal muscle behaved with heat. So if we put some hot water on the ventricles, then this will lead to higher amplitude of contraction with the same heart rate, right? Now if we put some cold water on the ventricles, this will mean that there will be lower amplitude of contraction with the same heart rate. Now till now we have experimented with heart in many forms. We stimulated the vagus nerve, we overstimulated the vagus nerve and got the vagal phenomena of vagal escape, went into the reasons of vagal escape, right? Like depletion of acetylcholine, etioventricular rhythm, etc. So now what are the other ways in which we can experiment with heart? Of course, one of the most fundamental ways in which we can experiment with heart is with the help of drugs, right? Now all of you must have heard of atropine. Yeah, atropine, the chemical which you must have used a lot during your internship, and all the people who are still doing their MBBS, they are going to use it profusely during their internship, and this is classically used in organophosphorus poisoning. In organophosphorus poisoning, if you see a classical patient of organophosphorus poisoning, what you will see, the pinpoint people, the people will constrict, and there will be lot of secretions, and there will be bradycardia, etc. like. So atropine is given for symptomatic relief, for immediately saving the life of the patient. The antidote of organophosphorus poisoning is pralidoxime, but atropine is given for symptomatic relief and for immediate saving of life of the patient. I mean, it will counteract all the effects of organophosphorus poisoning. And atropine increases the heart rate by blocking the vagus nerve, and it acts directly on the muscarinic receptors present on the heart. But even nicotine can increase the heart rate by acting at the site of ganglion. So how exactly we can know which drug has acted? I mean, you might get a graph in which vagus nerve is stimulated, and the stimulation of vagus nerve is not producing any effect on heart rate. So it can be either atropine, it can be either nicotine. So how do we know that which drug has exactly acted, is acting on heart? So this is very much like mechanical engineering. I mean, they give you a car, you remove a component and run the machine, and if the machine is faltering somewhere, that particular function is the function of the part which you have removed. So what we'll do is that we'll stimulate the vagus nerve at different points and just see the effect on the heart rate, right? So now we know that atropine acts directly on the heart. If something is acting directly on the heart, this means what? That stimulation of vagus nerve anywhere won't produce any effect on the heart, right? Now, suppose this is a vagus nerve, there is a ganglion, and this supplies the heart, right? Now, atropine is acting directly on the muscarinic receptors of the heart. Atropine is directly acting at this level, right? And blocking the effect of vagus, right? And nicotine acts at the level of ganglion. If you are experimenting with frog, this ganglion is called crescentric line, right? Crescentric line is situated just at the border of sinus venosus and the auricle, right? So basically, if you have given the drug atropine, no matter where exactly you stimulate the vagus, there will be no effect on the heart. But if you have given the nicotine, then if you stimulate the preganglionic fibers, there will be no effect on the heart rate. But if you have stimulated the crescentric line or the postganglionic fibers, then there will be effect on heart rate. Now this could come in exams as some kind of graphical representation, right? So how they will give a question of this in exams, right? This will be like this might come something like this. This is suppose the heart rate. Now you start giving vagal stimulation. Okay. As soon as you give vagal stimulation, strong vagal stimulation, the heart rate stops, and then the heart starts beating once again. After that, you give some kind of chemical. Let's call it X. Right? After that, you give vagal stimulation once again. Now, when you give vagal stimulation, nothing happens to the heart rate. It continues same way. But now what you do is that you give vagal stimulation at the level of CL. Right? You give vagal stimulation at the level of CL. That means you are stimulating the ganglion. If you are stimulating the ganglion and still nothing happens to the heart rate, that means you have stimulated both the preganglionic fibers, nothing happens to the heart rate, postganglionic fibers, nothing happens to the heart rate. Vagus has become completely ineffective. Then this means the chemical given is atropine, right? Now suppose we draw another graph, right? We draw another graph in which we stimulated the vagus over here. Now we draw another graph in which we give a chemical called Y. Now when we give vagal stimulation again, nothing happens to the heart, right? But this time when we give vagal stimulation at CL, the heart rate stops. The heart which was beating, it stops after we give vagal stimulation at CL. CL is ganglion over here, right? So if we give stimulation at CL and the heart stops, that means whatever chemical we have given is acting at the level of ganglion, and that might very well be nicotine, right? So physiology is one of the most interesting subjects which you are going to study in your MBBS, and even after passing your final year examination, even after doing your PG, you realize one of the subjects which you enjoyed the most during your MBBS course was physiology. So basically it's all about concepts. You don't have to memorize a lot. You have to just read few basic things, learn their interaction, and then you can answer a myriad number of questions, right? So hopefully you enjoyed this video. In case of any question, you can write on the comment section below. Wish you all the very best for your future. Take care. Bye bye.